Good evening, Vanshika. Good evening, Asma. Okay. So in the last class, we had discussed regarding the production of, you can say, the specific 17 beta estradiol, and how does that estradiol cause the ovulation, and how is that perpetuation goes on. Okay, so I'm Rahul Saha, and today we'll be discussing the spermatogenesis, okay, and also the fast block and slow block to polyspermy. This is a very, very important event when it comes to any NEET examination, right? People are not really aware of what is slow block and fast block to polyspermy, and sometimes they're unaware of the complete spermatogenesis event also, the in-depth one. So I'll give you a table first, okay? So spermatogenesis, I'll give you a specific table, you can note it down. So cell type, this side, then meiotic events, okay, then chromosomal complement, this is how many chromosomes and how many DNA, okay. These things you should be aware of, okay. So the thing is, we are aware of the fact that there are two types of spermatogonium. There is one thing known as type A, there is something known as type B. The type A spermatogonium is a stem cell type spermatogonium. It would divide via mitosis and generate one more type A spermatogonium so that its supply never ends, and one more type B spermatogonium. Okay, so it's the type B spermatogonium which will undergo spermatogenesis, type B spermatogonium. Okay, so now it will enter, you can say, the DNA replication. Okay, the S phase of DNA replication before meiosis. It will enter DNA replication stage, and when it does enter DNA replication, it will have 46 chromosome but 92 DNA. So 2n4C means each chromosome would have its sister chromatid like this. So 92 DNA, but chromosome number will stay 46 like this. 46 chromosomes would be there, but each chromosome would be containing its, you can say, sister chromatid. So therefore it will be 92 DNA. So good evening, Tarun. So that is how it goes on. Okay. So then after the meiotic event is over, okay, after the meiotic event starts here, commences here, it would become, after the first meiotic division, you can say meiosis 1, it will become primary spermatocyte. It would become primary spermatocyte. Okay, here no polar body production would be there, so first meiotic division is still in progress. Okay. Division in progress, and since it is in progress, it will still be, you can say, 2n4C. Now after this event, first meiotic division gets completed. Okay, now it will be over, the first meiotic division is complete. Now after this, you would be forming, you can say, two secondary spermatocytes. Okay, you can say two secondary spermatocytes. Here the second meiotic division is in progress. The second meiotic division is in progress. Now, once second meiotic division is in progress, the chromosome will become 1n, means haploid already, and then 2C, because DNA 23 chromosomes are there, but they are all in the, you can say, sister chromatid stage. Like we know already about how meiosis you can say commences, perpetuates, so it will become 1n2c. Now here we will have the conclusion of the second meiotic division. Second meiotic division completed. Now after this you can have, you will be producing 4 spermatids. Now you have produced 4 spermatids, okay, and then what we will be writing down under the, you can say, meiotic event is immature haploid gametes. Now haploid gametes are there, but they are immature haploid gametes. Now once they are immature haploid gamete, they will be all haploid, so 1n 1c. Now 4 spermatids will now undergo spermiogenesis. They will undergo spermiogenesis, okay, to produce 4 spermatozoa. 4 spermatozoa which are mature haploid gametes. Which are mature haploid gametes, and which are again 1n 1c. 1n 1c. So this is the overview of the spermatogenesis, or you can say sperm synthesis event. Now spermatogenesis, okay, it results into the formation of, you can say, or the production of 150 to 275 million spermatozoa per day in humans. Okay, 150 to 275 million spermatozoa can be produced per day in humans. Okay, so it's a huge number to be noted. Now it begins where? It begins in the seminiferous tubules of the testis. When we will be talking about the anatomy of testis, I'll explain it to you. The seminiferous tubules of the testis, okay, after the onset of puberty. Now we know that at the base of the seminiferous epithelium are various types of populations of spermatozoa, or spermatogonia I should be saying. At the base of the seminiferous epithelium, at the base of the seminiferous epithelium, there are various populations of spermatogonia. Like you have oogonia or oogonium, spermatogonium or spermatogonia, right? Now there are type A spermatogonia and type B spermatogonia. So type A spermatogonia, they represent the stem cell population. You can note it down. Type A, you can say type A spermatogonia. It represents the stem cell population. Stem cell population, okay, which will mitotically maintain its number throughout the life, which will mitotically or through mitotic division maintain its number throughout a man's life, throughout a male's life. Meaning that, what do I mean? The meaning is that the type A spermatogonia, type A spermatogonia, they divide via mitosis, means they differentiate via mitosis to generate one more type A spermatogonia, and in the other pathway they differentiate into type B spermatogonia. Okay, so this type B spermatogonia will take part or will delve into the pathway of spermatogenesis, whereas the number of type A spermatogonia got maintained. It remained constant as it gave rise to one type A spermatogonium per type A spermatogonium, so that the stem cell population pertaining to the spermatocyte, okay, pertaining to, you can say, the spermatogonium never ceases, never decreases. Okay, that is why a male, even if he is 90 years old, and if he is really healthy, non-diabetic, non-hypertensive, there is no cancer and all, then he can go for reproduction. Okay, he can still give rise to children. Okay, so but the females cannot achieve this feat because their oogonium doesn't, you can say, increase its stem cell number. They directly go towards, you can say, oogenesis. Okay, so they undergo or they face the process of menopause, where the menstrual bleeding stops, but we, the males, we never cease to produce sperm in our life if we stay healthy, right? Reproductively healthy, I should be saying. Then next is, there is a research in which it has been seen that type A somatogonia give rise to type B somatogonia, which are destined to leave the mitotic cycle and they enter meiosis. So entry into meiosis is stimulated by retinoic acid, a derivative of vitamin A. Means retinol is oxidized to retinoic acid. This is the carboxylic group, that OIC group, right? So retinoic acid, upon the stimulation of retinoic acid, this type B spermatogonium, it leaves mitosis stage and enters meiosis, that is spermatogenesis stage. But there has been some conflicting, like in this case retinoic acid works, but earlier they used to think that in every meiotic division the initiation is caused by retinoic acid. That research has been refuted of late, but as per NCERT, as per the NEET syllabus, just stick on retinoic acid as the precursor for meiotic division. But in all the cases retinoic acid does not trigger meiotic division as per the latest research. So that's the information that I would give you, but still tick on retinoic acid. Okay, old information, but still it is correct only as per the course. Okay, now all the spermatogonia, okay, are sequestered at the base of the seminiferous epithelium by interlocking processes of the Sertoli cells. Okay, Sertoli cells, they can release inhibin, they can release the androgen binding protein, they do release the anti-Mullerian hormone, right? So by the interlocking processes of these Sertoli cells means they will, you can say, emanate the processes through the cytoplasmic extensions like cilia, like this. So by the cell membrane processes only, it is like the processes of the Sertoli cells. Okay, you can call it microvilli like processes. Okay, now what happens is, these are complex cells which are regularly distributed. The Sertoli cells are complex cells which are regularly distributed throughout the periphery of the seminiferous epithelium and occupy almost 30% of its volume. Okay, and they are heavily involved. There you can say in time, you can say intimately involved in all the stages of spermatogenesis. Okay, and a single Sertoli cell, I'll tell you, can support from 30 to even 50 developing sperm cells. A single Sertoli cell, okay, nurse cell, Sertoli cell can support 30 to 50 developing, you can say, sperm cell. Sperm cells. Okay. Now, you can say that the molecular signals from a Sertoli cell determines whether a type A spermatogonium will continue through the spermatogenetic path or remain as a stem cell, or will it undergo cell death known as apoptosis. So the thing is, when type A spermatogonia, or let's start talk about spermatogonium, is divided into one type A, another, you can say, type A. Now this type A suppose becomes type B. Okay. Now who will decide it? Molecular signals from Sertoli cells. It will decide whether this will become type, it will stay as type A, it will become type B, or this type A will undergo cell death. Okay. Natural programmed cell death known as apoptosis. Okay. Apoptosis is a type of cell death where the DNA is degraded by the internal proteases known as caspases, whereas simple cell death is known as necrosis, where the plasma membrane bursts open. Okay, it bursts open. So this is apoptosis, that is programmed cell death, where DNA is degraded, you can say. So the Sertoli cells, they play a huge role, you can see. Otherwise, both can become type A, type A. Other type A becomes type B only through the signals given by the Sertoli cells, the molecular signals given or perpetuated by the Sertoli cells. Okay. Now, what happens is, you can say, the progeny of type B spermatogonia, it can complete the meiosis, the leptotin stage, the zygotin stage. Okay, but they are still, these cells, they stay interconnected. Okay, within the barrier of the Sertoli cell and the seminiferous tubules. Okay, the Sertoli cell processes, or the Sertoli cell microvilli processes, are very tightly joined to form an immunological barrier known as the blood testis barrier between the, you can say, forming sperm or synthesized sperm cells and the rest of the body. Okay, including the newly formed type B spermatogonia. So I'll write it down. The Sertoli cell, that's a very very important neat question. The Sertoli cell processes, okay, they are, you can say, are tightly joined, they are tightly joined, okay, tightly joined and form an immunological barrier, immunological barrier known as the blood testis barrier. Okay, between the forming sperm cells, sperm cells, you can say, and the rest of the body, and you can also include the spermatogonia and the rest of the body, including spermatogonia. Now just understand what I have written. The thing is, you know already that the immune system, our immune system, has got this intricate, drastic ability to discern, to recognize self from non-self, self antigen from non-self antigen. Okay, that's why we have to take drugs like immunosuppressants like cyclosporin, okay, Trichoderma polysporum, it's the fungus that produces it. So we have to take several immunosuppressant drugs when we go from, or when we go for, you can say, allogenic transplantation. Same species, different individual. Like if I go for kidney transplantation, if I take your kidney, then even if I match the blood group, even if I match the human leukocyte antigen, okay, which are present, which are known as the MSC class 2 molecule, still the minor HLA will not be matched, and my T cells, okay, will be the my cell mediated immunity, my T cells, my T helper cells specifically will be able to recognize your kidney which is inside me, and we recognize that the antigens present in that kidney are not the same as the antigen present on the rest of my body, and then it will mount an immune response on that transplanted kidney and will completely destroy that kidney, necrose that kidney, and entire kidney will be under acute kidney failure again, right? So that can also happen in a sperm. Now you will say sperm is produced in our body. Why would it happen in sperm? So how does a sperm get produced? So sperm get produced by meiotic division. In meiotic division, we have a process of crossing over or not. The crossing over generates variation, right? The crossing over will generate, you can say, chromosomal exchange. Okay, wait, I just so it will generate, you can say, chromosomal exchange. These arms will be exchanged in homologous chromosome in the Pachytene stage, and when the chromosomes are exchanged, you can say new proteins will be made or not, because genetic variation has occurred. Now they have switched places, so new genetic code could be there, new proteins could be produced, and that protein is not the same as the protein produced in your body. So the antigen produced by the sperms, the spermatozoa, at the after the spermatid has been transferred, or you can say it has developed and matured through the stage of spermiogenesis and become spermatozoa, it will produce antigens which are not resembling identically your antigen, which are not identical to your antigen, okay, your body's antigen. Now the if T cells can reach here through the blood, because they are present in the blood, if they can reach through the blood and reach the new spermatozoa, the just produce this spermatozoa, they will mount an immune attack on it and destroy the spermatozoa, and a man will become infertile. Same case will happen in case of ovum. So you have the blood ovum barrier also that I'll discuss too, okay, later on. So we don't want any T cell infiltration where spermatozoa has been produced in the seminiferous tubules. Therefore, the seminiferous tubules are completely guarded, completely sealed by the tight junction produced by the processes of Sertoli cells. Tight junctions are produced by proteins like occludin, claudin, you can say claudin. Okay, so these are nothing but cell adhesion molecules. The cells are adhered very, very tightly like this. Okay, there is no gap for the paracellular transport, like this. Tightly they are adhered. So paracellular transport cannot happen. Only receptor mediated transcellular transport can happen through the cell, not between the cell. Between the cell transport is known as paracellular transport. So they don't give any area for paracellular transport. On top of that, the microvilli will be surrounding the microvilli of the Sertoli cells, or the simply known as the Sertoli cell processes, will be surrounding it, and the Sertoli cells will be tightly packing it so that other cells cannot come in. Only nutrients can come in through diffusion. Okay, and transport mediated active transport and all, Na plus and all, they can come in, vitamins can come in, but the cells itself, the T cells, cannot reach, and therefore our newly produced spermatozoa Therefore, they stay protected. They stay safeguarded from the attack which could be mounted if the T cells, the T helper cells can reach through the blood onto the site of spermatozoa production, that is the seminiferous tubules. Therefore, it is known as a blood testis barrier. So, the blood cannot get through it. Only nutrients can go through via transportation or receptor mediated transport through the channels and transporters via the transmembrane proteins, not blood, right? Not the T cells. It is known as the blood testis barrier and is perpetuated by the Sertoli cells. Very, very important. Okay. Now, the progeny of the type B spermatogonia, which have entered the first meiotic division, are known as the primary spermatocytes. Okay. Now, located in a characteristic position just inside the layer of spermatogonia and still deeply embedded in Sertoli cell cytoplasm, the primary spermatocytes, it spends almost 24 days passing through the first meiotic division. Okay, so primary spermatocytes, it requires almost 24 days to complete the first meiotic division. Okay, first meiotic division, that is known as the primary spermatocyte. Okay, now during this time, the developing sperm, that is the primary spermatocytes, it uses a strategy similar to that of the egg. What is the strategy? Producing in advance molecules that are needed at later periods when changes occur very rapidly, meaning that it will involve the synthesis of a variety of RNA molecules and storage of RNA molecules. Storage, you can say synthesis and storage of RNA molecules. Okay, so the thing is, RNA transcription is a very, very active process from the early stage of spermatogenesis to the time when spermatids begin to form. So you need to keep a lot of mRNA, rRNA, tRNA molecules with you. One more thing I'll tell you, which is extremely important for NEET examination, is that thing known as protamines. Okay, so a well-known example of preparatory mRNA synthesis involves the formation of protamines. You can say preparatory, specifically preparatory mRNA synthesis is the formation of protamines. Very, very important. You can underline the word protamines. Protamines are what? They are small arginine rich and cysteine rich proteins. Very, very important. They are arginine rich. First, you can write small. They are very small polypeptides. Okay, they are very small arginine rich and cysteine rich proteins. Okay, cysteine rich proteins that replace the lysine rich nuclear histones that we have already learnt about histones, right? In the molecular basis of inheritance chapter, I taught you. So that replace the lysine and arginine rich, especially lysine are more. So lysine rich, you can say nuclear histones. Now, why will the histones be removed? Because histones, they compact the DNA, they compress the DNA. We have talked about the, you can say the solenoid model, beads on a string model, then 300 nm, 700 nm, then 1400 nm, chromosome model also 300 nm, chromatin model also 10, 11 nm, you can say then 30 nm models. So why would the histone be removed or replaced? It is not that they want it to become DNA, because if the histones are removed, it will become a 1 meter long DNA, right? So we don't want it. But protamines, what they do is they remove histones. Why? Because it allows higher degree of compaction of nuclear chromatin required during the final stages of sperm formation. Because higher degree, understand it very very carefully, of compaction is required, you can say, at the eventual stages or final stages of sperm formation. Sperm formation. Meaning that the compaction which is attained by the help or assistance of stones is not enough, is not sufficient. You need a higher degree of compaction. So that higher degree of compaction can be perpetuated by, can be caused by the arginine rich and cysteine rich. Cysteine is a sulfur containing amino acid. Cysteine and methionine and sulfur, two only, these are the two only amino acids which contain sulfur, can form disulfide bond, can form disulfide linkage. So it is replaced by arginine rich and cysteine rich, which forms the disulfide linkage and can cause more compaction compared to what can be attained by the action of histones. And more compaction is required at the eventual stages of sperm formation, because you need to, you can say, accommodate more and more RNA. So you need to compact the DNA so that the cytoplasm can accommodate organelles also and lot of mRNA also. So you need to further compact or further compress the nuclear material so that the nucleus takes the least space, as less as possible, least space possible. Okay, theoretically possible in the final stages of sperm production. There is a spermatid, then spermatozoa. Okay, so these protamine mRNAs are first synthesized in the primary spermatocytes, okay, but are not translated into proteins until the spermatid stage. So, one more very very important answer of the question: protamine mRNA. When does the synthesis begin? Just tick on the option primary spermatocyte. But its translation, okay, into protamine protein does not begin in primary spermatocyte stage. It begins when the sperm has reached the spermatid stage. Okay. Now from spermatid to spermatozoa, when it will mature, when it will be differentiating, then compaction will be more and more and stones will be replaced. Okay. So what happens is, in the meantime, the protein mRNAs are complexed with proteins and are inaccessible to the translational machinery. Okay, so they are complexed with different proteins so that they cannot bind to the ribosome, then IF1, IF2, IF3 cannot come in and they cannot initiate translation because they have to be inaccessible to the translation machinery. Okay, now why so? Because if protein mRNAs are translated before the spermatid stage, what will happen is the chromosomes will condense prematurely and then the sterile sperms can be produced. Okay, that is the reason, because everything has a reason. So if protamine mRNAs are not complex with proteins and made inaccessible to the translation machinery, means if protamine mRNAs are translated before the, you can say, stage or before the stage of spermatid formation, then it will result into formation of premature compaction and sterile sperm synthesis. So that will occur. Okay, sterile. It will result into formation of it will result into, you can say, generation of, you can say, premature compaction and sterile sperm synthesis. So that is why before the spermatid stage, okay, before this underlined spermatid stage, the protamines are never, you can say, translated. They are never ever translated before that. Okay, so always keep that in mind, and then we can proceed to the next topic, like increasing numbers and kinds of small RNAs. Okay, they have been seen in every type of sperm production. Okay, so that has been seen in sea urchin also, in human beings also. Now we can proceed. So after the completion of the first meiotic division, the primary spermatocyte gives rise to two secondary spermatocytes, okay, which remain connected. Always remember, it may not be given properly, but which remain connected by a cytoplasmic bridge. So, after the completion of the first meiotic division, okay, the resulting state is what? The primary spermatocyte gives rise to, you can say, two secondary spermatocytes. Two secondary spermatocytes which remain connected. Very, very important stuff this is, which remain connected by a cytoplasmic bridge. Okay, which remain connected by a cytoplasmic bridge, because this concept is always hidden in many books. So you should know this concept. They are connected by a cytoplasmic bridge. Now the secondary spermatocytes, they enter the second, you can say, meiotic division without delay. Like the primary spermatocytes, it took, you can say, 24 days to complete the first meiotic division. It will enter the second meiotic division without delay. Now this phase of meiosis is very, very rapid. Okay, and gets typically completed in approximately 8 hours. It doesn't take 24 hours. So it enters meiosis 2 without delay and only takes 8 hours to complete the division. Only takes 8 hours to complete the division. Now each secondary spermatocyte would produce what? Would produce two immature haploid gametes, which are known as which are known as spermatids, right? So each secondary spermatocyte would produce two immature, okay, haploid gametes after the completion of meiosis 2 to produce, you can say, two spermatids each. So in total, 2 plus 2, total number of spermatids will be 4. 4 spermatids would be produced. Okay. 2 plus 2 spermatids, 4 spermatids will be produced from two secondary spermatocyte. Okay. Now the 4 spermatids produced now will then start to translate their protamines. They'll form protamines. They'll replace this arginine rich and cysteine rich protamines will replace the lysine arginine rich histones and cause more compaction of the DNA. Okay, and the 4 spermatids produced from a primary spermatocyte progenitor, okay, are still connected to one another. They are still connected to one another, okay, and typically to as many as 100 other spermatids as well. They are still connected, okay. So spermatids do not divide further. What happens is they undergo a series of profound changes, you can say, that transform them from ordinary looking cells to highly specialized spermatozoon. The plural is spermatozoa. Now they'll go specialization, okay, differentiation, no division, okay. So I can call them, even differentiation is a division. So they'll undergo specialization, specialization to become sper, four spermatozoa. Singular is spermatozoon. Okay. Many times people are not aware of this singular form. Singular, it is known as the singular form is spermatozoon. Okay. Now the process of transformation from spermatids to spermatozoa is called spermiogenesis or also spermatid metamorphosis. We can say the process of transformation, okay, from spermatids to spermatozoa is known as spermiogenesis or spermatid metamorphosis. Spermatid metamorphosis. Okay, so always keep that in mind because this is spermatid metamorphosis. This is not mentioned, but sometimes in competitive examination, just to perplex you, confuse you, trouble you, they'll ask what is spermatid metamorphogenesis. Spermatid metamorphosis. So metamorphosis means the formation of all the organs. So differentiation and specialization only. So just you will tick in the competitive examination that spermatid to spermatozoon or spermatozoa formation is spermatid metamorphosis or spermiogenesis. Okay. Now the next step is several major, okay, categories of change occur during spermiogenesis. Okay. One is the progressive reduction in the size of the nucleus and tremendous condensation, you can say, of the chromosomal material, okay, that I've already told you. This condensation happens. Why? Because I'm reiterating it, because of the replacement of stones by the cysteine rich, arginine rich proteins, small proteins known as protamines. But along with the changes in the nucleus, also it has been seen that a lot of reorganization, you can say profound reorganization, occurs in the cytoplasm. Okay, now what happens is cytoplasm streams away from the nucleus, okay, and a condensation of the Golgi apparatus also occurs. Okay, so you can say first of all protamines will cause, you can say, supercondensation of the nuclear DNA. Also, in case of cytoplasm, in cytoplasm, the Golgi apparatus, okay, the Golgi apparatus at the apical end of the nucleus ultimately gives rise to the acrosome. Okay, the Golgi apparatus at the apical end of the nucleus, okay, gives rise to acrosome. Very important competitive exam question. From where does Golgi apparatus gets, or sorry, from where does acrosome arise? That is Golgi apparatus. Now what is acrosome? The acrosome is an enzyme filled structure which plays a very crucial role in the fertilization process. That is known as the acrosomal reaction. Okay. Now what happens is, at the other end of the nucleus, a prominent flagellum grows out of the centriolar region. At the other end, okay, you can see the various specialization processes which are happening in a simple round spermatid to make it spermatozoon, right? The sperm cell. So at the other end of the nucleus, what happens is, we have a prominent growth of flagellum from the centriolar region. The centriolar region, I can say, causes the prominent growth of flagellum. Growth of flagellum. Okay. Next is, the mitochondria then get arranged in a spiral around the proximal part of the flagellum. Proximal means the near the flagellum. You can say proximal is near, distal means far away. So the mitochondria, they're arranged in a spiral manner around the proximal part of the flagellum. The mitochondria are arranged in order to provide it enough amount of, you can say, GTP, okay, are arranged in a spiral manner around the flagellum, because the flagellum is what? The flagellum is nothing but they are microtubules made up of alpha beta tubulin dimer. Okay, both are attached to GTP. Okay, but the GTP attached to the beta subunit is hydrolyzable. Okay, to here more alpha beta units can be attached. This one is sterically hindered GTP, which cannot be hydrolyzed. So this is the minus end. The growing end is the positive end. It has a specific polarity. You might have studied the cytoskeleton in class 11th. The microtubules, the microfilament, and the non-polar micro, you can say, cytoskeleton known as intermediate filament like desmin, keratin. Okay. So after this, what happens is, during spermiogenesis, the plasma membrane, you can say, of the or present at the head of the sperm is partitioned into several antigenetically distinct molecular domains. Okay, now these domains, they undergo numerous changes as the sperm cells mature in the male, and at a later stage what happens is, when the spermatozoa are traveling through the female reproductive tract also, okay, they make more and more antigenic changes which help them in the long run to avoid, you can say, immune response, and to circumvent the immune response. In that sense, you can think of it like that. Now as spermiogenesis continues, The remainder of the cytoplasm, which is known as the residual body, it moves away from the nucleus. Okay, it moves away from the nucleus. The remainder of the cytoplasm of the cytoplasm is known as the residual body. Okay, what happens to it is shed off. Okay, it is shed along, you can say, the developing tail of the sperm cell. Okay, it is shed along the developing tail, okay, of the sperm cell. One more thing I'll tell you is when it is shed in vesicles, this cytoplasm, extra cytoplasm, and these vesicles are then phagocytosed by, you can say, the Sertoli cells. These vesicles are phagocytosed by the Sertoli cells. Okay. Now, on completion of spermiogenesis, which takes approximately 64 days after the spermatogenesis process, okay, it takes almost, you can say, 64 days to complete spermiogenesis. Okay, 64 days means since the inception of spermatogenesis to your completion of spermiogenesis, it takes almost, yeah, or you can say it takes 64 days. Spermiogenesis, 64 days to complete. You can say the spermiogenesis means from the advent of spermatogenesis to the completion of spermiogenesis. I am talking about that. Okay, not from spermatid state to spermiogenesis. I am talking about from spermatogenesis, okay, to spermiogenesis complete. 64 days it takes. Okay. Now the spermatozoon now has become a very highly specialized cell, well adapted for motion because it has great flagellum and the delivery of its packet of DNA to the egg because now it has the acrosome, it has the mitochondria which is surrounding the, you can say, flagellum. Now the sperm cell consists of the following. Okay, it will contain a head which is almost 2 to 3 micrometer wide and 4 to 5 micrometer long. Okay, so now the sperm or spermatozoon contains the following. It will contain a head, okay, which is 2 to 3 micrometer wide and you can say 4 to 5 micrometer long. Then it will contain, you can say, the head also contains the nucleus and acrosome. Okay, contains the nucleus and acrosome. Number two, if I say the sperm contains, you can say, after head comes the mid piece, right? Comes the mid piece. The mid piece, okay, what it does is the mid piece contains the centrioles and the proximal or the initiating part or the part which contributes to the start or initiation of the flagellum, okay, and the mitochondrial helix which surrounds the entire, you can say, flagellum. Okay, so it contains the centrioles, the proximal part, proximal part, of the flagellum and the mitochondrial helix, I'm calling it because that is a cluster of mitochondria surrounding the flagellum. Okay, helix. And then I can talk about the next part of the mitochondria, which is the tail. Okay, the tail is about 50 micrometer long. So the tail is about 50 micrometer long. Okay, and consists of a highly specialized flagellum. Okay, highly specialized flagellum. Highly specialized flagellum. Now I'll show you one thing, okay, where you will be able to notice that a sperm always, okay, there is a latest research, okay. Earlier it was thought that sperms, they spin, or you can say not spin, okay. So what happens is they swim, right? Sperms swim via flagellar movement, but a latest research has happened which has shown that the sperms, they spin and not swim. They just tumble down and they spin, okay, and not swim. This is a great research which has been done in 2020. So more than 350 years, the scientists were fooled by the meaning that the flagellum, it will move in a zigzag way and the sperm is swimming through, okay, the female reproductive tract, but no, it causes the tumbling motion and it spins, okay, through, and the spinning motion causes the sperm to, you can say, propagate in the female reproductive tract. Okay, so I'll send the video also, the sperm swimming. Okay, so understand that that is the latest information regarding the sperm movement in the female reproductive tract. So I hope you are able to comprehend the lecture. So in the next class, we'll be talking about the capacitation of the sperm. Two types of capacitation. One happens in the epididymis, one capacitation. The capacitation part 2 happens in the female reproductive tract, and then we'll be talking about the fast block and slow block to polyspermy as well. Okay then. Thanks a lot. Let's